Hi, today I'm going to conduct an experiment about osmosis with shriveling potatoes. To start the experiment, label the paper H2O and label the other side H2O plus salt. Label the disposable bowls the same way. Then, get your cup, your water, and your salt. Put water in the cup and add salt into the cup until it's saturated. Saturated means that no more salt can dissolve in the water. You'll know when this happens because even after stirring it a lot, there will still be salt crystals at the bottom of the cup. Next, get your potato, your knife, and your cutting board and cut the potato into thin slices. You'll only need 2 thin potato slices for this experiment. Next, measure the diameter of each potato slice and write these measurements down. Next, place 1 potato slice on each bowl and put water on one bowl and water with salt on the other. Wait for a few hours to observe what happens next to the potato slices. After a few hours, I'm now back to measure the diameters of the potato slices again. You'll notice that the potato slice that was soaked in salt water is smaller and softer than it used to be. How did this happen? Well, the potato is made up of millions of plant cells, each surrounded by membranes that are semi-permeable. This means that the molecules can travel into and out of the cells, especially water, which is a solvent. While the potato is alive, the cells can decide which chemicals they want inside and which they want kept outside using special gate mechanisms in the cell membrane. But now that the potato has been removed from the ground, those gate mechanisms do not work as well anymore. If the potato is dead, they won't work at all. Instead, the chemicals surrounding and within the potato cells move in such a way that there's a balance inside and outside the cells. In the bowl filled with salt water, the environment external to the cells has a very high concentration of salt, a lot higher than the environment inside the cells. Solvent chemicals including water will naturally move in such a way that the concentrations of the solution are balanced. In this case, the water moves so that the salt is the same concentration inside the cell and outside the cell. The way this is achieved is that the water from inside the cells flows out through the membrane into the salty solution in the bowl. The water acts as a solvent and dilutes the salty solution, making the salt concentration outside the potato less salty and makes the solution inside the cells less dilute and more concentrated, or simply more salty. As the water leaves the cells of the potato slice shrivels up, and our measurements shows this. You might also see the slice in the clean water grow larger, as the water flows in the opposite direction, into the cell in an effort to dilute the other chemicals within the potato cells themselves. What you have just looked at is osmosis. This is the name of the movement of solvent molecules such as water through a semi-permeable membrane into a region of where there is a high concentration of chemicals other than the solvent, in this case, the salt. The movement of the solvent into the salty solution equalizes the concentrations of both sides of the semi-permeable membrane. For the vocabulary of this experiment, solvent is the material that dissolves a given substance, usually a liquid but can be a gas or solid too. Solute is the substance dissolved in the solvent as described before. In this science experiment, it was the salt. And that was my experiment about osmosis with shriveled potatoes.